Good morning, everyone. I'm sorry you were going to have a lovely blonde speaker from the Silicon Valley, but instead you're just going to get me, I think. So I'll try to keep you awake as much as she would have. So I'm going to talk about some of the research that we're doing. I don't really work with big data. I work a lot with small data. Sometimes we try to mine big data, but we're really interested in rare variants. So this is actually an issue because it requires even larger data sets. These are the different groups that we collaborate with to try to get to the numbers that are required to reach statistical significance, and it's a large translational project involving many individuals who have expertise from the genome to transcription, all the way to the neuroscience and the clinical phenotype. So I'm going to talk about neuropsychiatric disorders and their genetics, and what we've been working on a lot are copy number variants. So the research that has been conducted in the past 5-8 years has revealed a daunting heterogeneity in neuropsychiatric disorders. So it's something that we somewhat expected, but not to the point where I think we've reached today. So this is an estimate in the latest paper using the Simon simplex cohort on autism, showing that depending on the type of variant, we expect somewhere between 600 and 1,000 genes involved in just autism. And so if you extend this to other neurodevelopmental disorders that are overlapping with autism, we're well above the 1,000 genes involved. So this is a little bit daunting, but also exciting. And so there's very practical steps that we need to take to try to tackle this problem. And what we've been looking at from a very practical perspective are copy number variants. And why are we looking at those copy number variants is that they represent a particular type of these variants that are recurrent. So the exact same mutation, structural mutation, occurs over and over in the general population. And they're maybe not as rare as some of the other mutations that are found, so the single nucleotide variants that are found in these neuropsychiatric disorders. So this is a copy number variant, so you can see, let's see, how does this work? I'm colorblind, so I can't see. Oh yeah, here we go. So this is what you are, normal diploid, so you have 2 copies of your autosomal genes, and then you may have at certain regions deletions or an excess of genomic material, which is a duplication. And the structure of the genome is such that on certain chromosomes, this is chromosome 16, the deletion and the duplication occur with the exact same recurrent breakpoints. If you look at all the copy number variants that are identified today and clearly associated with autism and schizophrenia, the 2 large neurodevelopmental disorders that we're interested in, here's a series of them, and together these represent almost 2% of the general population. So they're rare mutations in themselves, but cumulatively they're rather frequent. And their effect is pretty large. So here you have an example of the effect. So this is data looking at all copy number variants associated with neuropsychiatric disorders in the Icelandic population. About 1.5% of this population carried one of these neuropsychiatric copy number variants. So this is the general population score on these different cognitive scores. So general population is 0, and then if you see this is the individuals who have schizophrenia, regardless of their mutations, and they lose about 1.5 standard deviation on their IQ. And if you look at the individuals who carry the copy number variants, who are associated with psychiatric disease, but do not meet diagnostic criteria for schizophrenia, so these would be the controls, but carriers of copy number variants, they do still have a very significant stigmate, a neurodevelopmental stigmate, with a loss of almost 1 standard deviation on their IQ. So these copy number variants are present in the general population, even when they don't reach, they don't lead directly to a neuropsychiatric diagnosis, they still have an impact on neurodevelopment. So the region that we've been working on a lot is this region here on chromosome 16. It's a region that it has many segmental duplications, and these particular structural duplications lead to large deletions or duplications in this region that occur in about 1 in 2,000 individuals for the deletion, 1 in 2,000 individuals in the duplication. So about 1 in 1,000 individual is born with a deletion or a duplication in this region that was initially associated with autism in 2008. And we started looking into this region in this rearrangement, trying to understand how it really affects neurodevelopment, and this is results from cognition in individuals who carry this variant. And the model is a very simple additive model. So here you have the normal distribution of global intelligence in the general population, and this is a theoretical effect of the deletion if it takes out about 2 standard deviations without any type of interaction. And this is the actual data that we have, and it really does show a very simple shift in IQ without any type of visible interaction. So it's a very additive effect, and basically the deletion is introduced in the family, and you can almost sort of predict the outcome by subtracting 29 points to the familial IQ. We are also very interested in energy balance in the context of neuropsychiatric symptoms. So obesity is one of the main comorbidities, is actually one of the top comorbidities in neurodevelopmental disorders, in autism in particular, and in schizophrenia. And antipsychotics don't help because they add on another layer to this. So this is actually the first patient I saw with a 16p deletion that was referred for severe obesity and some problems of learning disabilities. And at the time, we had identified back in 2009 a deletion on 16p, but we didn't really know what it was. And we started looking into the effect of this rearrangement on obesity. And so as I said, this comorbidity is very important with neuropsychiatric disorders. And one of the hypotheses is that the factors that are altering neurodevelopment, this alteration of neurodevelopment lead to neurodevelopmental symptoms or psychiatric symptoms, as well as an alteration in energy balance. So we looked at the association of this deletion with issues of energy balance, and we looked into case control GWAS studies that had been conducted in the general population on obesity, comparing individuals with obesity to individuals the general population, and we identified a few of these deletions in these populations, and we also collected additional cases of the deletion in general population cohorts. And basically, we did a very simple case control study calculating the frequency of the deletion in obese individuals compared to the frequency of the deletion in lean or overweight individuals. And there's about an odds ratio of 40, so a huge impact of the deletion on the risk of developing morbid obesity. So this was the result of this first study, showing that about 0.7% of all individuals with morbid obesity in the general population carry this deletion. And if you present with morbid obesity and some symptoms of neurodevelopmental disorders, you have about a 3% risk of carrying this allele. So at the time, we were interested in this because it was one of these examples of a rare variant that was responsible of a common disorder. And today, I think that the debate is still open on what is the contribution of rare alleles to common disease, but it's probably a lot higher than what we initially expected. We were also interested in understanding how this region actually modulates satiety and energy balance. And we took a look at the opposite rearrangement, which is the duplication. If I can show it here, so the duplication here. And with the question of what does the duplication, what effect does the duplication have on a BMI if we know that the deletion is associated with a very, very high risk of morbid obesity. So we had the same strategy. We collected all duplication cases that were identified in general population cohorts and obesity cohorts, and then also in neuropsychiatric cohorts, and we collected a series of these individuals showing that the risk of being underweight was much higher in the duplication than in the general population. So here we have now our updated data set showing that the duplication, the intrafamilial controls, and the deletion represent three distributions showing the impact of this region on modulation of BMI. You have other traits that follow also this mirror effect of modulation on a simple trait, which is head circumference, which is a proxy for brain volume, and I'll discuss this a little bit later. What's really actually interesting is that this copy number variation is not followed by any type of compensation at the transcription level. So if you have a deletion here, and this is work that we did in collaboration with the CIG, Alexandre Raymond and Lausanne. If you have the deletion, you have low transcriptional levels of all genes comprised in the 16p11.2 region. If you have a duplication, the level of transcription is higher than the controls, which are in blue. So none of the genes are compensated, and you just have a correlation of this transcription level with the number of copy number variants. So there's this interesting situation with copy number variants is that if you do look at traits among individuals who carry duplications or deletions, you can identify actually mechanisms or dimensions that are correlated to the copy number variant, and here we did it for BMI, but we've also looked at brain volume, which also follows this phenomenon. Now there's another very interesting aspect that has been observed in the past maybe 5-8 years with the discovery of all these new genes associated with neurodevelopmental disorders, is the fact that the same genes have been identified in patients with autism and in patients with schizophrenia. Not all genes are overlapping, but a large amount of genes seem to be associated with both disorders, and several hypotheses have arose. Some of them may be suggesting that these two disorders are actually overlapping with normality here, and then when you when you depart from normality, you are at risk of developing either autism or schizophrenia. But then there's other evidence that suggests that maybe normality is in the middle, and then sometimes some mechanism, if they're pushed in one direction or the other, may lead to these two opposing disorders. So this is still a working hypothesis. It might be true for some regions, some genetic regions, but not for all, but this is it's a sort of a paradigm that we're studying now. So today it's difficult to study this hypothesis because we don't really have a measure a quantitative measure of schizophrenia that we could also apply to autism. In fact, there's no quantitative measure of either autism or schizophrenia. There's no quantitative measure that shows that if you are above 2 standard deviations, you will have a high risk of developing autism or schizophrenia. So this is sort of a hypothesis that is very difficult to directly apply at the clinical level. So then you can start looking for what we call intermediate phenotypes. And in neuroscience, we're not as lucky as some of the other MDs and other disciplines. We really don't have access to the brain, and we really don't have access to much other measures. One of the measures that we do have access to now, which is a little bit cumbersome, is neuroimaging. So we did use neuroimaging, and so this is a preliminary data set that was recently published in Molecular Psychiatry, showing that if you look at the way this 16p region modulates brain structure, and if you look at very simple metrics like total intracranial volume or total gray matter or white matter volume, you see that if you have a deletion, a duplication compared to intrafamilial controls, all these measures are correlated to the number of genomic copies. So the region just seems to very simply modulate in a very simple matter this metric. And we also, in collaboration with Katsani, show that this region can modulate brain volume in animal models such as zebrafish, also by just manipulating one of the genes, KCTD13, which is one of our top candidates. If you ask the question in an unsupervised manner, what are the regions in the brain that are structurally modified and modulated by this region, you can see here that the statistic, this is sort of like a whole brain association study, a BWAS in some way, where you look at each little different region of the brain, which is a voxel, and you ask, is this region of the brain correlated to the number of genomic copies? And what the algorithm tells you is that this region here, the reward system in some areas in the temporal region, as well as the projections, the white matter projections connecting these regions are associated with the number of genomic copies. And this is actually a network which replicates very well the reward system, which is implicated in obesity, but which is also implicated in autism and schizophrenia. So it's possible that the genomic region alters one brain structure then, which leads to 3 different types of symptoms that co-occur in the patient. So beyond copy number variant, we wanted to see if actually individual gene expression levels would allow us to elucidate the individual contributions of these genes to these structural, neuroanatomical structural variation. And in this study, we were very underpowered, so it's more of a proof of principle, but it seems that it's something that's possible. So here you have the 18 genes that are our top candidates within the 16p11.2 region, and you see here for each gene the individuals who have duplications, so you have a wide distribution of increased expression. You have the individuals who have a deletion who have low expression, but you already see here that the number of genomic copies already gives you 70% of the variance of gene expression of the genes within that region. So you're really only working with this additional 30% of variance. So a larger sample size will be required, but in this proof of principle approach, what we show is that, for example, one of our top candidates in this region, MAP kinase 3, if you plot the expression level across all groups, you see that the putamen, which is in the striatum, one of the key regions that we think is implicated in these patients, the volume of the putamen is correlated to the gene expression level, which was measured in lymphocytes in these patients. So the previous talk, I think it was very clear the power of these biomarkers, and in neuroscience, it's even getting access to circulating peripheral biomarkers is even more crucial because we do not have access to the organ in patients, and we will never, almost never have access to it unless there's a major technological breakthrough that will allow us to observe functioning brains in all these patients. So for us, developing biomarkers that will give us an insight on the mechanisms and their relationship with the clinical outcome is very important. So this is sort of another little story that's related to these neurodevelopmental disorders, autism and schizophrenia, is that what we see in the clinic is, and this has been observed for many, many years, is that for every girl that is referred to us, 2 or 3 boys are referred to us. So there's an excess of males who develop psychiatric and neurodevelopmental symptoms, and this is even more striking in autism, where, for example, in high-functioning autism, it's almost 5 males for 1 female. So this is a piece in The Economist that was done after this paper here that we published last year, showing that investigating molecular signatures of this sex bias doesn't really help us exactly understand what's going on, but it gives us an idea of how the causal mutations may interact with the sex in the development of these neurodevelopmental disorders. So what we did is that we went back to a clinical database, and this was unfortunately a private company, Signature Genomics in Washington, the state of Washington, which had a large database of 15,000. Now they have 30,000 patients that they have investigated for copy number variants, but they are not linked to the medical records. So we weren't able to extract the exact symptoms of all these individuals. The only thing we know is that they were referred for neurodevelopmental disorders and autism. But what you see is that this is the odds ratio of the number of mutations in females over the number of mutations in males. So if it's 1, it's the same amount in both groups, but as you rise above 1, that you see an excess of mutations in females. So as you rise to more deleterious mutations here, which are larger, so this is 400 kb, 1 megabase, you see that you start seeing more and more mutations in females that are referred for neurodevelopmental disorders and autism. And here, if you look at just other metrics of deleteriousness, which is rare and large copy number variants, you really have an excess of mutations in females, which you see here with another metric, which is the fact that they're rare, they're large, and they occur de novo in these patients. So here you almost rise to about 1.4 in terms of odds ratio with a very significant p-value of 10 to the minus 11. You see the exact same thing in the parental transmission, and here this is the ratio of maternal to paternal mutations, and the same thing as you rise in the deleteriousness of the mutation. When it's transmitted in almost 60% of the cases by the mother and only 40% of cases by the father. Same thing in another independent cohort of the Simon simplex on 841 families, you see the same phenomenon. And this really suggests that the same factor here, the same genetic factor, seems to be less deleterious in females, and they tolerate it a lot more without demonstrating symptoms. So before they're referred to the clinic, they really have a much higher burden, a much higher mutational burden. So just to finish, I talked about just a few regions, and what we're seeing now is the incredible heterogeneity, and this is going to be a massive problem. So if we're actually deconstructing these diagnostic categories into tiny, rare disorders, there's no way we're going to be able to tackle a treatment or development of treatment and development of care for these patients. So one of the movements today and the hypothesis is that hopefully all these different genes that are causal when they're mutated, causal for autism, may cluster in pathways, and these patients, although they don't have mutations of the same gene, might have a common denominator that may be targeted with new treatments. And there's different ways of looking at these pathways. This is a very classic way of looking and clustering genes into pathway based on protein-protein interaction. And I think there's pathways that are really now emerging, such as chromatin remodeling, synaptic function, and the Wnt signaling pathway. Here's a different approach. This is a paper that is in press that we worked on with our collaborators in Lausanne, showing that the 16p region, which is a very strong predisposing factor to autism, has chromatin contacts with a series of other genetic regions, and you can design pathways this way also and show that this pathway, which is a 16p genomic disorder related pathway, in fact highly overlaps with autism pathways, and that it may represent a subtype or a subgroup of ASD. Eventually, what we would like to do is start understanding if we can regroup patients into functional, relevant groups, and then we could apply a drug discovery in these groups. And this is one example. In neuroscience, this is I can't see my pointer. This is a key molecular mechanism regulating synaptic homeostasis. It's the mechanism regulating protein synthesis at the synapse. And there's a series of genes here that are regulating this molecular mechanism. And all of these genes, when they're mutated, are responsible of genetic disorders. So here you have Fragile X, which is very well known as a genetic form of autism. You have tuberous sclerosis here with TSC that directly regulates mTOR, which is implicated in this pathway. And then here you have 16p11.2, which is deletion or duplication of ERK1, which is a direct regulator of protein synthesis at the pathway. And there are actually compounds that can restore the alterations in this pathway in these disorders in animal models. And in 2008, we launched a clinical trial, several clinical trials based on about 8 years of preclinical data showing that in Fragile X, where the absence of FMRP dysregulates this mechanism, you can restore this in the animal model. And after about 6 years, the conclusion in humans is that there's absolutely no effect at all. So it's not that I don't want to finish on a very negative note, but I think that at least in neuroscience, discovering genes is clearly one of the only ways today to get insight on mechanism. The mechanism will give us insights on new drug targets, but it still might be a very tough way before we actually find treatments that we're able to apply early enough to make a difference in these patients who have probably an alteration, a very, very early onset alteration, or even a prenatal onset alteration, even in disorders such as schizophrenia, of their brain structure and organization. So with that, acknowledgements and thanks for your attention.
Questions, please. I have a quick one. I was just wondering, the obesity variants that you are picking up, whether they have impact for the general population.
So say that again.
So the variants that you're picking up, they're sort of linking obesity and psychiatric disorders. I'm wondering what is their contribution when you look at the general obesity population?
So So you can look at the explained variants in different ways. Here, so for rare disorders, the explained variance in the total general population is small because these variants, individually these variants are rare. But the variance explained in the individual that is carrying the genetic variant is very large. At a population genetics level, the variance explained is small, yes. But these variants can be used in the clinic because for an individual who carries this variant, the effect size are 2 standard deviations, for example. So these are very large effect size in the individual, and they have a very high predictive value.
Just a really simple question. The gene expression data that you showed, what tissue is that coming from?
So the gene expression data showed it was lymphoblastoid cell lines. People, as always, complained a lot about trying to infer a neurobiological mechanism based on expression levels measured in patients on lymphoblastoid cell lines. But I think really today there's enough data accumulating that at least for certain pathways, this is really a relevant approach. And surprisingly, many of the genes that are involved in these complex neuropsychiatric disorders are very basic processes. So it's a lot of cell growth, a regulation of protein synthesis, and you can actually measure the alteration. For example, the protein synthesis alteration, you can measure it in lymphocytes. It's altered in lymphocytes, it's altered in skin. You can have a plus 200% increase in these measures, but there's just no phenotype in these tissues. So the tissues tolerate this, but the brain probably doesn't. So we think that at least for certain types of mechanisms, you can measure it peripherally, and it gives you probably a good indication of the basal alteration in the brain tissue. At least that's what we hope.
Thank you very much.